Our protocol describes the use of a hydrogen peroxide biosensor in cultured zebrafish neurons and larvae. This protocol will help researchers to dissect the role of reactive oxygen species in normal physiology and pathophysiology.
The main advantage of this technique is the real-time detection of hydrogen peroxide in living animals and culture cells. This method can be applied to other animals like rodent model system.
To prepare the cover slips, use acid-cleaned cover slips stored in 100% ethanol. Use forceps to remove one cover slip from the storage container, and flame it to remove residual ethanol. Air dry the cover slip completely by placing it at an angle inside a 35mm culture dish. Prepare poly-D-lysine, or PDL, working solution. Apply PDL to the center of each cover slip, avoiding spreading of the solution to the edges, and incubate the PDL for 20 to 30 minutes at room temperature. Make sure the PDL does not dry out. Wash the PDL with 0.5 milliliters of sterile water, and let the plates dry completely. Prepare laminin working solution, and apply it to the center of each cover slip, making sure to avoid the spread of solution to the edges. Incubate the plates at 37 degrees Celsius in a humidified incubator for 2 to 6 hours. Avoid a drying of the laminin solution. To perform embryo dissection and plate retinal ganglion cells.